Hello everyone, welcome to the new video tutorial of Bio and Tech. In the last session, we have dealt about cell biology. We have discussed the introductory part of cell biology. We have also discussed the colloidal nature of cytoplasm. In this session, we will be dealing with the techniques used in cell biology. That means we will be discussing the various scientific principles and techniques used in cell biology and also instrumentation mainly seen in cell biology. If you haven't subscribed to our channel, subscribe to it and enable the bell icon to get daily notification. So without wasting time, let's get on with the video. Okay, so techniques that we have to discuss in that which is commonly used in cell biology includes the first one is microscope. This is the portion that is really important and most frequently asked questions from this portion. So the instrumentation portion is microscopy. Other three are mainly the techniques that use the scientific techniques used in cell biology. The second one is flow cytometry. Third one is FISH, that is fluorescent in situ hybridization, and the fourth one is DISH, that is genomic in situ hybridization. Now we have to discuss all these one by one. So first let's focus on microscopy. The first one, that is the microscopy, is used to magnify living objects. If you are seeing in the biology perspective, is for living things, to magnify microscopic level things to our naked eye. Microscope is mainly used. Now we have to see the different properties and principles that are used in microscopy. The first one is magnification. Of course, it has to magnify the objects, right? So the magnification equation is size of the image with microscope, that is size of the image that we see through the microscope divided by the size of the image with naked eye. So that gives the magnification of a microscope. So magnification is important principle or part of microscope. The second one is resolving power. Suppose you think that there are 2 points which is closely placed together. Now while viewing to the microscope, it should be seen as 2 points. So that perception is very important. So the ability of a microscope to see these 2 points as 2 separate points is known as resolving power. Now another term that is related to resolving power is the limit of resolution. That means limit of resolution means the distance, minimum distance between these 2 points to perceive it as 2 separate points is known as the limit of resolution, and the equation of it is 0.61 lambda by Na, where lambda is the wavelength of light and Na is numerical aperture. Now the relation between limit of resolution and resolving power is that when limit of resolution decreases, resolving power increases. It is inversely proportional. So a microscope should have high magnification and high resolving power. That means it should have lower limit of resolution. So the numerical aperture of microscope is almost fixed. Now what is changeable? The lambda, that is wavelength of light, can be changed. So a lower wavelength of light should be used for higher resolving power and magnification. That's an important point to be noted here. The third one is contrast. A high contrast image makes it more easy to understand its components or the components of a cell or microscopic living organisms. So that is very important, that these are the 3 points that we have to relate to a microscope. Now the next thing that we have to focus here is the types of microscope. We have to deal with the different types of microscope in brief to understand the basic concept of these all microscope. The first one is simple microscope, then compound microscope, phase contrast microscope, and the fourth one is interference contrast microscope, fluorescent microscope, UV microscope, electron microscope, confocal microscope, dark field illumination microscope. So these 9 microscope should be studied in brief to understand the basic concept, how the working principle of these microscope should be studied, so that we can answer some of the basic questions in the entrance. So now let's focus on the first 2 in this class, that is the simple microscope and compound microscope. Now the first one, that is simple microscope, it is usually used for the dissection, for seeing the LS of a flower or dissection of an organism in zoology. So in botany it is usually used for the LS, for seeing LS of a flower in taxonomy. That is the main use. It has a single lens system. Now it may not be microscope as you may imagine, it can be a single lens. A simple lens can also be known as simple microscope. So it just used to magnify what is there in our hand or what the object we have to magnify. So the main point related to simple microscope, it has a single lens system, and the image when you see through the lens will be upturned. The upturned image will be seen when you see through a simple microscope, and the magnification is usually 4 to 40 times. So these are the important points or important concepts or basis of simple microscope. Now when you're seeing the parts of a simple microscope, the first one is a foot, that is the foot which helps the simple microscope to stand, and the stand, there's a stand in which the vertical limb is attached, and you can see a folder down where the convex lens is seen. So convex lens, not biconvex lens. A convex lens is attached. It can be foldable, can be movable, and there's a glass stage for the light to come to the object. So there's a glass stage and there's a mirror for reflecting the light, and two clips are there for attaching if we are seeing a slide for lower magnification, two clips will be there for that, and adjusting screw for adjusting magnification for focusing the object. So that are the three parts of a simple microscope. You can see the image in the right side. Okay, so that's all about simple microscope. Now let's focus on the compound microscope. Okay, now compound microscope. Now the first point is not related to compound microscope. Generally saying about microscope, the first attempt of learning microscope was done by Janssen brothers. But the first microscope with multiple lens system was developed by Robert Hooke. Now coming on to the compound microscope, nowadays compound microscope has a 3 lens system. The first one is condenser lens, the second one is objective lens, and the third one is ocular lens. But the basic magnifying of image that is done by the 2 lens system, that is the objective lens and the ocular lens. So actually the compound microscope is a 2 lens system, but for actually viewing purpose, so for comfortable viewing, the 3 lens system was introduced. That is, the first one is condenser lens, which helps in gathering the light and focusing into the object. It involves a diaphragm where which we can adjust the amount of light that should enter the object. Now the objective lens first magnifies the image and it produces an image, magnified image. Now the image which is magnified by the objective lens becomes the object of ocular lens, and it again magnifies it. So it actually 2 times magnification occurs. Now the magnification of compound microscope is, suppose it should be multiplied. The magnification of objective lens is to be multiplied with the magnification of ocular lens, and then you get the total magnification of compound microscope. Suppose it is the objective lens has 5x and ocular lens has 45x, 5 into 45. That's the total magnification of a compound microscope. You should actually multiply the magnification power of objective lens and ocular lens, then you get the total magnification power of compound microscope. Now the entrance related points, which is very important, is that the resolving power of a compound microscope is 0.2 micron. That is an important point. You should never forget that. So that should be noted down. The other structure of a compound microscope is not that important, but I have given the picture there. You can learn, look into and understand the basic structure of a compound microscope. So that's all for today's video. If you have any doubts or confusion, please mail to bio@gmail.com. Wait till next video. Bye.